Well, first, let me begin by thanking you for the opportunity to speak here tonight. It's stuff that I'm deeply excited and passionate about. So the title of my talk is called, and can you folks hear me? Great. It's called Decoding the Brain. And really what I hope to convince you about in the next 20, 30 minutes is that the brain is not a black box. The brain is something that we can know and understand. And that I think we're at a very exciting time right now where we're just starting to learn some of these principles. So to begin with, first, just about what I'm going to be talking about. First, I'm going to give you a brief historical overview of how we've come to understand brain function. Some of the dynamic analysis and applications of some of the recent things that have gone on, some of their implications, and some of the future directions. So really, when you look at it 200 years ago, the first ways that we began to understand brain function incorrectly was through phrenology, basically ascribing function to different bumps on the head. And I think it really started to take on some level of seriousness in the mid-19th century, where basically with lesions in the brain, we started to learn about how different parts of the brain work by its dysfunction. And kind of a classic example is Phineas Gage. He was a railway worker who basically was a very God-fearing, very serious man who basically got a railway spike through his brain, which you kind of see right here. And it was kind of very newsworthy back then. And he went, and it basically injured the frontal lobes of his brain. And after that, he became a very disinhibited gentleman, where he would cuss all the time. He would kind of go to the bathroom on the streets. And really, people at that point suddenly realized, well, the frontal lobes are important for personality and inhibition. And that was kind of one of the first forays in kind of essentially the study of lesions of the brain. And really, that went on for really the next 150 years, and even to somewhat today, where we still learn about how the brain works by certain areas of dysfunction. So, and kind of concurrent with that, really at the turn of the century, we learned that not only that the brain is an electrical organ, that it generates electrical activity. And this gentleman, Hans Berger, out of Germany, was the first guy to discover EEG. And even at that time, he thought that, well, maybe that these oscillating brain waves can tell us something about how the brain works and even understanding thoughts. But it really wasn't until really kind of the 80s where we started to look at some epiphenomena associated with brain activity, which told us about where function was occurring. And it was usually with kind of such things as alterations in blood flow, blood oxygenation, or metabolism. And we used those changes to identify what certain areas of brain function, where they were located. And again, they gave us kind of some rough pictures of where activity in the brain was going on with certain cognitive functions. And just to give you a brief kind of summary of those, for instance, we learned that the back part of the brain, the occipital lobes, are involved with visual processing. Again, a lot of these things we learned through lesional studies, but this is really the first time that we were essentially seeing the brain in action. The frontal lobes, let's see if coming up here, yeah. Again, involved in executive function, higher level decision making. Inhibition, as we learned with Phineas Gage 150 years prior. Our temporal lobes, kind of the bottom part of our brains, and the middle part of those temporal lobes involved with memory. The back part of our frontal lobes involved with motor function. And two areas known as Wernicke's area and Broca's area, again, which we initially learned by lesioning, involved in the production and the understanding of speech. But it's not perfect yet, because in some ways we're still ascribing function to lumps and bumps. Now we're doing a little bit better of a job that now it's actually on the brain. But so it really was, I think, with animal models, again, in the 70s and 80s, where we started to really kind of where we started to learn how information is encoded in the brain. And really with some of the monkey models in the 80s, and I would like to go through just a brief example of that, where when they started putting microelectrodes into the cortex of brains, where they started to learn how the brain is actually encoding information. And just to give you this is one of the first examples of that, a kind of a famous gentleman by the name of Georgopoulos. And he did a fairly simple experiment. He had electrodes in the monkey motor cortex. And what he had the monkey do is he had it manipulate this thing called a manipulandum. Basically a simple articulated arm. And the monkey, and you can see his arm right here, basically had to do what's called a center-out task. And what that involved was basically moving from the center of the screen here to some peripheral target, which you see circled. And here's actually the tracings that the monkey drew out. And you can see it's at 0 degrees, 90 degrees, 180 degrees, 270 degrees. And when you actually looked at the neuronal firing rate, what they basically found was that for a certain direction for that particular neuron, it had essentially a directional preference. And when you, not to get too involved in this, but when you regress that to a line, it formed a cosine curve. And they started to refer to this as cosine tuning, meaning that basically for a given neuron, it had a directional predilection. And that when it fired at a rapid rate, that would predict the monkey's arm movements. And when you looked at a whole bunch of neurons together, you could actually predict very precisely in three-dimensional space where that monkey wanted to move his arm. And this is just a kind of a graphical representation of that. And each of those lines represents the action potential firing rate. And it's kind of like a bunch of neurons voting, and the loudest guy voting seems to kind of predominate. So again, looking at this, you can kind of appreciate some of the implications for this for somebody with the severe motor disabilities. Now, so that was single unit systems. Now also, again, going back to these electrical rhythms from the scalp, again, we were able back then to get very simple signals for whether the motor cortex was active or not active, depending on how these certain frequency bands changed in amplitude. And so, again, we could use that as a signal for very simple device control. And let me show you an example of that. Oops. And again, this guy has a, oops. Can you see my mouth? Oh, I guess not. Here we go. Oh, keeps him disappearing. But he's got an electrode cap on. Those are, again, recording signals from the surface of his brain. And he can control the cursor, whether it goes right or left, based on motor imagination. Got to work at it a little bit here, but eventually he gets it. So now I've shown you some good examples, but really getting to the next step as a clinical platform, something that can actually help somebody with some severe motor disabilities, there's some problems. And part of that is with EEG and single unit systems, they have features to them which really prohibit practical application. EEG gives you relatively simple control, and because it has problems with signal to noise, it gives you somewhat erratic function. It really takes a long time to train to get very simple levels of control. Single unit systems, as I've shown you, they get very high level of control, but they're relatively invasive. You have to actually put them in the cortex. And also, and this is kind of very clinically relevant, is that scar starts to form around those very small electrodes. And because of that, they lack what we call durable effect. They only last for around 6 months in monkeys and around 6 months in some preliminary human trials. And what that means is that you have to do kind of repeat surgeries every 6 months. And that really isn't practical for somebody with some severe disabilities where they have to keep on relearning how to use this. And where kind of our group at Washington University came into the picture was that it was an alternative strategy, was using what's called electrocorticography, or ECOG. And that's signal acquired from the surface of the brain without penetrating the brain. And that signal has some unique features in that it's much more robust than EEG. You can see a much broader frequency. It has much better regional discrimination, meaning that you can really see kind of fine points of activation, millimeters versus the centimeters with EEG. And because you're not penetrating the brain, scarring is much less of an issue. This is actually kind of a picture for, hopefully nobody's too faint of heart here, but this is actually a picture of a human brain during surgery. And we actually put these grid electrode arrays for a clinical indication for localizing seizures. There's a certain population of patients with intractable epilepsy actually require the placement of these electrodes over the surface of the brain so we can find where those seizures are coming from and then actually take that part of the brain out to treat their seizures. This also provides us with a very unique opportunity to essentially get invasive electrical activity from the brain. So these patients have these electrodes over the surface of their brain for a couple weeks while we wait for them to have seizures. And during that period of time, we actually do experiments with them where we have them participate in various cognitive tasks. And what we found was, and kind of really somewhat to our surprise, it was kind of walking into this Aztec gold mine where we suddenly found all this really cool information was that where EEG really gave us only frequencies of around 0 to 40 hertz. When we started to look at these frequencies under the skull, there's kind of a whole world opened up. These high frequencies. And what we started to discover is that these high frequencies carry substantial information about cognitive intentions. And this is just a brief example. When we looked at, for instance, patients moving a joystick around, kind of like that manipulandum that I showed you earlier with the monkey moving it around in circles, was that depending on which direction they moved, these high frequencies were able to tell us what direction the human being was intending to move. And so, for instance, when they moved in this very simple example, looking at one electrode over motor cortex, when we looked at frequency over time, that when they moved to the left, the high frequencies here would go up, and when they moved to the right, those high frequencies would go down in amplitude. And so we started to find multiple different electrodes that could essentially help us predict which direction the person wanted to move that cursor. And we put that to some good use, and I'll show you some examples. Again, we found some signals which told us about cognitive intention, and now we wanted to utilize those for device control. And this is an example of one of our subjects who is controlling a cursor, again, with thought alone, towards 4 peripheral targets. And can you see that cursor? It's kind of small. And also, just to put this into perspective, so he did this in about an hour of training. And some of the other, relative to some of the other constructs with single unit systems as well as with EEG, it takes around, with single units it takes several months, with EEG it takes several months to years to get even close to that type of control. So the control was very fast, and part of that was predicated on the fact that we were utilizing these high frequencies. No, there's nothing connected to it. This is all from brain activity alone. Yeah?
So what is he thinking about there?
It's a great question. Yeah, so the question was, what is he actually thinking when he's doing that? Well, initially we tell him, imagine moving your hand going up, down, left, right. But and that's what he originally was doing. But when you ask him at the end of the session, he says, well, and again, this is anecdotal, I can't prove this, but he says, well, I just wanted to go up, down, left, right. It becomes intention-based. And really what we found is, and I don't have the slides in here to show it to you, but people, very quickly, once they get this closed-loop feedback, basically learn to tune their brain signals to what the computer is looking for. And they learn it very quickly with feedback.
I noticed the subject moved his head.
Uh-huh.
Can you have that sort of effect with just a brain movement? Just through moving the eyes rather than moving the head?
Oh, the head movement actually has nothing to do with him. He's just following it. That's all. It's purely brain activity. That it really, and I'll show you some other examples here to show you that that's actually kind of not important. So here's another example where he is moving he uses actual movements this time, which you're going to see. He's going to play Space Invaders here. And to get the cursor to go to the right, he uses real hand movements. But again, it's being derived. The control is being derived from brain signal alone. Right hand movements move his cannon to the right. Tongue movements, which you can't see, move it to the left. So he's got basically 2 control features, right and left. So you can see he's basically waiting for that final Space Invader. He shoots it. And if you watch his hand, he's actually really good at dodging and juking. And again, this is brain activity that's controlling this. So he's kind of upset about that. Now, but one of the important things is that essentially when you look at brain activity, especially with motor movements, there's essentially very little difference between real movements and imagined movements. So now we say, well, you really don't actually need to move your hands. Stop that. Pretty much he has the same level of performance. But I think, one thing that I want to kind of, talk about is this kind of notion of the neural code, or kind of somewhat of a colloquial term. But I think what what it's telling us is that really, we kind of really found the iceberg with brain-computer interfacing. And the point being is that the information in the brain is knowable, that we can decode this information, this electrical activity from whether it be cortex or even deeper brain structures, to really understand the information of our thoughts and our intentions. And so, and kind of more strictly defined, the idea being that there's fundamental electrophysiologic rules which govern kind of information in the brain. Again, layman's term, the electrical language of the mind. That the information of our thoughts is coded by some defined electrical patterns. What are some of those patterns? And that I think just as to draw a parallel, I think it's almost kind of like the human genome, is that just as the genome is governed by information coded in a series of nucleotides, that there's a correlate level of organization, and we're just basically just beginning to scratch the surface of that. And so the way that some of this information is encoded is in spatial encoding, what part of the brain is involved, and temporal encoding, meaning, for instance, action potential firing, as I showed you earlier with the firing rates of neurons, as well as frequency encoding, meaning frequency band and how those frequency bands change. And I think, just as I think, certain kind of, movements in science have really led to some things that have changed our society and changed the way we live, such as, lunar exploration and kind of, research in space really led to, computers, internet, modern jet propulsion, satellite communications, modern materials, really from the 60s leading to now. And mapping out the human genome has really led to kind of, a revolution in, novel molecular techniques for treating diseases, infections, cancer, biotechnology, bioinformatics, individual risk assessments, personalized medical therapies. I think that understanding the way that information is encoded in the brain has that same level of gravitas. That basically the possibilities for more efficient ways of communicating between people, seamless human-machine interactions, novel treatments for psychiatric and neurologic disorders, and ultimately, if we really understand how the brain encodes information and processes it, I think it really gives us a better chance for creating better models for artificial intelligence. And since we're here talking about telecommunications, I think that if we're improving the way that information is relayed, that I think that there's a real correlate relationship between how culture and technology evolve with the rate of information transfer. For instance, the age of reason and the scientific revolution and the age of enlightenment were really predicated by the printing press. That, in the mid-16th century, when they suddenly created the printing press, suddenly information was disseminated at a much faster rate. So people started to think more and learn more from the discoveries that people were making. And that really kind of transited through the ensuing centuries, that with the evolution of modern countries and infrastructure that supported postal systems, the telegraph, and the phone, that you see kind of a matching level of increase in technology. And I think that today with the internet, there's an explosion of information, and technology is moving almost at a dizzying pace. And I think that hopefully in the future, as these techniques of improved information transfer rates with the BCI technologies, that we'll see that even more dramatic changes in technology. And so let me close with, just to give you a sense for how quickly technology moves, I want to tell you about Pong. And this is what they said in the 70s. This is from Time. 7 quarters later, they were having extended volleys, and the constant Pong was attracting the curiosity of others at the bar. Before closing, everybody in the bar played the game. The next day, people were lined up outside Andy Capp's at 10 a.m. to play Pong. Around 10 o'clock that night, the game suddenly died. The machine coin container was full. That's in the 70s. People were lining up to play Pong, thinking this is the most amazing thing. When you think about it, now 30 years later, these video games are commonplace. They don't even attract our attention. We certainly wouldn't line up outside of a bar to play a video game. And I think that in 30 years, it's been a dramatic change in what we're seeing in technology. And I think that, kind of thinking about it now, we're essentially playing the equivalent of Pong right now with brain-computer interfaces. Any questions? Yeah, back there in the blue shirt.
Well, you were reading motor information out of the motor cortex and impulses. You went to the sensory part, and have you looked for impulses there? And have you ever thought about, or have you ever tried putting some information back in, seeing if somebody could actually see or hear something?
No, I haven't, but people are doing that. Certainly haptic feedback and especially visual prosthetics, if something has been a very active field that people are involved in, ranging from stimulation anywhere along the visual axis, from your retina to your optic nerve to actually to your visual cortex. And there are people out there actually who have experimentally implanted brain electrode arrays placed over their occipital cortex, that back part of the brain, to stimulate vision. People who are completely blind. And they're able to see what we call phosphors, or kind of bright flashing lights with these stimulation, and are actually able to utilize this construct to maneuver around a room. So that's definitely happening. Yeah.
Where are the cones? Where are the colors that the cones receive mixed? I'm thinking of impressionist paintings.
Well, as far as the colors that we see, where are they processed? So just to repeat the question, she's asking where are the colors that we see mixed. And now you're starting to kind of go beyond some of my kind of expertise, which is more optic physiology. But the information is initially processed in the eye, where you have rods and cones, as you mentioned. A lot of that information goes to the back of the brain, and there's what are called ocular dominance columns, which first process points of light, and then those points of light are further synthesized in other columns, which then are bars, and they're what are called ocular blobs, which are in cortex, which have a certain processing for particular colors. Now, as far as how exactly that processing has occurred, I'm not sure is entirely clear at this juncture.
So it's not in the nerve?
Part of it's in the eye, but also part of that processing goes on in the brain. In the brain, that's right. Not so much in the nerve itself, no. Yeah?
Thank you for your presentation. It's exciting. Is any of this have any implications for the ability to download the contents of the human brain with a series of computer files? Say, is there anybody thinking about that, or is any of this work trending in that direction?
People have done certain things like that in mice, actually, where in the so one part of our brain called the hippocampus is involved in memory predominantly, but in particular spatial memory. So, for instance, like if a mouse is navigating through a maze, they monitor its firing rates, and basically there's very selective firing patterns for a given path that it takes. And you can actually take that information out and know kind of which direction the mouse has gone in. And you can actually replay, and when the mouse is sleeping, you can actually listen in again, and the mouse actually replays it backwards. So that's kind of indicative of learning that's going on while you sleep. So the question is, how long do these electrodes stay in? Now, for these patients, this research is piggybacked on top of a clinical therapy. So they stay in for 1 to 2 weeks. But the other, I think, kind of getting at your question is, how long can they stay in? And that's a question which we're investigating right now in monkey models. Now, people have done research in the past in other types of animal models, and they've shown that these electrode constructs can stay in for over a year without a degradation in signal. So essentially, you're asking about bit rate, like how much information can you transfer, and that's essentially in the brain-computer interface and neuroprosthetics worlds. That's kind of the kind of benchmark of kind of it's almost the intellectual arms race, where we always want to push and see how much information we can get out, so that not only are you doing one-dimensional control is a certain bit rate, two-dimensional control, say XY axis, is another kind of level of bit rate information transfer. Then you get to 3 dimensions, and then multi-dimensions. And right now, with electrocorticography, we're at 2 dimensions. We're working towards 2 dimensions with a click, so that you can select. In the single unit systems, they're at 4 dimensions now, meaning you can move in 3-dimensional space and grab. And I don't think we've defined what the information limits are yet, but we were continually able to kind of, with evolving computer speeds and signal analysis methods, moving up and up and up. So I don't think we've defined our limit yet. So the question is, have people gone beyond the fact that just action potential firing rate as far as information encoding? And certainly there's numerous ways that the neuron encodes information, not just in its firing rate, but in terms of the connections that it makes, what type of neurotransmitters it's releasing, what type of neurotransmitters it's receptive to, and how those other neurotransmitter receptors activate it or inhibit it. There's a lot of work going on in that front. And one of the things that people have really learned is that, for instance, memory and the way that in our responsiveness is certainly affected by kind of what we call synaptic plasticity, like how responsive it is and how to certain transmitters. Do we know, for instance, like the kind of the CG, A, and T of how information is coded? I'd say we're probably at maybe like CG, but I don't think we have all the letters yet. We certainly know, for instance, that action potential firing rate, frequency, location, and how those frequency change all are responsible for encoding information. And we've been very successful at utilizing those letters to essentially provide types for certain cognitive intentions, ranging from motor function, such as motor direction, as well as, for instance, we're starting to decode phonemes, basically components of words over speech cortex. We're able to get at that. So, but I don't think we have all the letters yet, but I think we're getting there.
Do you foresee a time when this fellow mentioned downloading, when we can actually upload thoughts one to the other person?
Assuming that we have, again, I don't think it's impossible. I think that theoretically, I think we're very much in the early stages right now. Right now, we're at decoding primarily. I don't think we're at the level of encoding, although people with visual cortex and auditory stimulators are starting to do a little bit of that. But it's the early stages of that.
We are anonymous. We are legion. We do not forgive. We do not forget. Expect us.